Okay, so hi everyone. My name is Nicole Marone, and myself and Coral Marina will be your new seminar coordinators for the year. I just want to quickly say thank you again to Annie and Alyssa for doing such a great job with the seminar. And with that, I want to introduce Justine Sauvage. So Justine Sauvage received her PhD in oceanography at the Graduate School of Oceanography, University of Rhode Island, under the guidance of Professor Stephen D. Hunt. Sauvage sailed as an inorganic geochemist on 2 IODP expeditions focused on unraveling Earth's subseafloor microbial world, its extent, diversity, and metabolic landscape. After her PhD, Sauvage started a postdoc at the Department of Marine Sciences, University of Gothenburg, Sweden, where she applies her background in oceanography, biogeochemistry, and ecology for development of integrated and sustainable solutions for the management of marine resources. Current projects include optimization of microalgae cultivation systems as a key component of a sustainable circular aquaculture industry, the development of functional aquifers, and the implementation of aquaculture components within integrated food production systems. So during this presentation, Dr. Justine Sauvage is okay with asking questions during the presentation. So please just raise your hand and we can unmute you, or you can also type your questions in the chat and we will read them as we get them. All right, Justine, it's on to you.
Thank you. Thank you so much for this introduction. And thank you for the invitation. So today's talk will be really about some of the work I did during my PhD with at the University of Rhode Island, as well as with many other colleagues, some at UNH and at BU and other places. And so yeah, the focus of this talk will really be to constrain radiolytic production of oxygen, hydrogen, and hydrogen peroxide, but I'll go through water radiolysis, but that's a process I'll discuss later, and really assess the relevance of this process to sustain microbial ecosystems in subseafloor sediments. So next slide. My slides are not moving. Any input? Oh, here we go. Okay, there we go. So before we go, I'll first would like to introduce that process of water radiolysis. So it's really the decomposition of water molecules after interaction with radiation. So very simply, when alpha particles, beta particles, or gamma ray interacts with water molecule, they initiate an ionization process and an excitation process, and which gives rise to a whole suite of intermediate reactions, but ultimately leads to the stable production of molecular hydrogen, oxygen, and hydrogen peroxides, as well as a bunch of short-lived chemical species like radicals. Now, this process has been known for many decades and has been studied in pretty simple systems like pure water or in the context of radioactive waste management, but no one has really looked at that process in terms of simply quantifying radiolytic production of chemical compounds in subseafloor sediments or in the ocean. Now, fundamental parameters of this process are really the yield. So how much hydrogen, for example, is being produced, and those yields are usually expressed in molecules hydrogen per 100 electron volts of radiation absorbed by the medium. And those yields are really a function of the radiation type. So is it alpha radiation, beta, or gamma, and also the irradiated medium. Now in homogeneous systems, so for example, pure water, those yields are extremely well constrained and well known. So they're depicted here for the 3 types of radiation. And you can see that for alpha particles, you have a pretty high yield, and that's really mostly due to how energy is being deposited in the medium. So for alpha particles, those particles really deposit most of their energy in a very short path. So it's really a pretty compact, compact path and process yielding to like more molecular products, while beta and gamma radiation also yield chemical compounds, but at a lower rate. So now in non-homogeneous systems, for example, subseafloor sediment, where you have a lot of pore water and then also sediment grains, the yields of radiolytic compounds and really focusing on hydrogen and oxygen are absolutely unknown. And some like systems have, no, some not systems, but some studies have looked at like the inclusion of like one mineral in aqueous solutions, like looking at the irradiation of slurries, for example, pyrite. And they saw that as soon as like a mineral compound was present, those hydrogen yields were enhanced. So there are definitely some like mineral-driven catalysis of water radiolytic production happening. Now this is kind of important because like most systems in nature are like not homogeneous, but like contain some like mineral or like some geological compounds. So really constraining yields of radiolytic hydrogen production and oxidant production is like fundamental for like a bunch of reasons, whether it's like management of radioactive waste, and for my own personal interest, like whether it's like to understand subseafloor microbial metabolism. So like I said, so hydrogen and after like water radiolysis really produced both like reduced compounds, so hydrogen and oxidized compounds like oxygen and hydrogen peroxides. And from a standpoint of microbial metabolism, that's pretty interesting because you're really producing both your electron donor, so really like the fuel, the food, and your electron acceptor, which is the oxidant to like oxidize the food. So that process of water radiolysis really produces both metabolites required for like microorganisms to like derive energy from like those molecules, like to perform redox reactions. So it's a pretty like win-win process for microbes. So with all this, like the three main questions of our study was to first really quantify radiolytic hydrogen production in subseafloor sediments to like see if we see those catalytic processes, and like just simply quantifying what's the production of radiolytic hydrogen in the subseafloor, which is like completely unexplored. Then the second question would be, once we know how much hydrogen is being produced, well, how does it compare to another like source of like microbial fuel, which is like, let's take the biggest like primary production in the overlying ocean water, just to have like a comparative idea of like the importance of those two processes relative to each other. And then really, so that's like really like on a global scale. And the third question is like more subseafloor microbial metabolism focused, and really like assessing what's the relevance of water radiolysis in terms of like sustaining microbial ecosystems. So those are the three questions I'll walk you through in this talk. And so the first question, so how much hydrogen is being produced in the subseafloor, so in like marine sediment, and is it like higher than in a pure water system? And I'll already give you the answer, and it's that all marine sediment types catalyze radiolytic hydrogen production. Now, to come to this finding, we did like a global survey of different marine sedimentary lithologies. So for that, we like collaborated and participated in a variety of scientific ocean drilling cruises, both through IODP and UNOS. And so you can see here the selection of 16 sites where sediment cores were extracted. And you can see that those sites were really are really dispersed across sediment lithologies found on the subseafloor. So really trying to capture like abyssal clays, biogenic sediments, but also organic rich continental margin sediments. So those are the 16 sites. And from those 16 sites, we selected 20 samples. So about 11 abyssal clays, because abyssal clays are really like your main, like at least in surface, your main lithology found across the seafloor. We also picked a bunch of biogenic sediments, both siliceous and calcareous in nature. Also some marl, so really like a mix of clay and calcareous ooze, and also a few lithogenous, so like continental margin sediments. And we always like picked grain sites that was like still pretty small, as that's like a fundamental factor in the water radiolysis process, because your sediment grains really need to be pretty fine, because that's where the radiation is originating from. So like if you have a fine sediment grain, some of that radiation can make it to the water to interact with the water molecules and produce that hydrogen. So you really want to be in a like fine-grained environment. So you can see some picture of those cores on the left, like those more biogenic sediment cores, and on the right, like a pretty like classical abyssal clay core. Now with all the samples, we performed a series of radiation experiments at the nuclear reactor on the University of Rhode Island campus, and we performed both alpha particles irradiation experiments as well as experiments with gamma rays. So separately, always in duplicate, we would like irradiate like sediment slurries from those 20 samples, and as well as 2 vials with just pure water, which we know the hydrogen yield from like many studies. So like I said, it's like very well constrained. And we really compared like production in water, production in sediments at different like irradiation doses. So really like to like kind of have an idea of like, is that a linear progression? Like the more you irradiate it, the more hydrogen you produce or not. To also have an idea of like, is it a linear production or like really constrain like how much the production regime. And so you can see on this graph, so on the y-axis, you have hydrogen concentration and dosage. And you can see that it's like a pretty linear trend. So the more you irradiate it, the more like hydrogen you produce. So we did those irradiation experiments, and then we measured hydrogen produced in each of those vials through gas chromatography. And by doing those like experiments, we could really like pretty accurately and constrain production yields of hydrogen for those different samples, so those 20 samples, and also test reproducibility, and all of that lined up. So this like ultimately resulted in like this summary of results. So on the left, you can see hydrogen yields for alpha particle irradiation, and on the right for gamma ray irradiation for all the lithologies studied. And the little dotted line on the verticals are really like your factor increase in hydrogen production in marine sediment relative to pure water. So really like showing like that catalytic effect of the marine sediment minerals. So you can see for the alpha particle irradiation experiments, so on the left graph, abyssal clays have a pretty high like hydrogen production yields, especially compared to factors. So it's like between a factor 13 to up to 27 higher than water. So a significant increase. And while others, lithology, so the biogenic samples and the lithogenic samples are like also like elevating hydrogen production compared to pure water, but to a lesser extent. But still, you can see that every single lithology examined did enhance the hydrogen production. And the same can be said for gamma radiation, with again like abyssal clays increasing yields by a factor 3 to like 5. But here, interestingly, it's really the siliceous ooze that like had the highest impact on enhancing hydrogen production, with up to a factor like 9 increase compared to pure water. So marine sediment definitely have like a catalytic effect on radiolytic hydrogen production, and therefore, how do you say, understanding of how much like hydrogen is actually being produced has been like in the past extremely underestimated, because so far only pure water yields were utilized. So that was the first piece of information. So now, once we know that, it's like on a global scale, how much hydrogen is being produced in marine sediments. Like we can like attempt to like calculate that, and then compare that also on a global scale to how does it compare to like primary productivity in the ocean. So how much like carbon being produced versus how much hydrogen being produced. So, and again, we're like asking this question from like, how to say, a perspective of microbial metabolism, because like organic matter is like pretty much like the paradigm, like essential like feed for like microbial communities, and like hydrogen is like the one we're like kind of like investigating its importance. So like really taking those two metabolic processes, so really the oxidation of organic matter versus the oxidation of radiolytic hydrogen to like generate energy for the microbes, because those like two metabolism like are like, yeah, it's like a different reaction. So if you want to really directly be able to compare them, it's like important that we work with the same currency. So really how many electrons are being transferred by like performing this metabolic reaction. And so that's why we're really, every calculation I'll discuss further is all expressed in electrons mole transfers per mole organic matter oxidized or per mole hydrogen oxidized. So that's just to like facilitate the comparison between those two processes. So to really like get to a global budget of radiolytic hydrogen production in marine sediment, I collaborated with different colleagues from the USGS, BU, and also the International Ocean Discovery Program. And we really like used a formally described model of water radiolysis that really like calculates based on a few inputs, and those inputs are radioactive element content of the sediments, hydrogen yields for the 3 different radiation types, and also physical properties of the marine sediment. Now, Blair et al., like based on those 3 inputs, like proposed a pretty solid like method to really like calculate hydrogen production. So to apply that, now that we have like revised hydrogen yields based on our experimental data, we can like use global data sets of seafloor lithology, seafloor sediment porosity, and sediment thickness to really kind of like apply that Blair calculation on a global scale. So that's what we did. So we used seafloor sedimentology data set from the Naval Oceanographic Office. And so for every lithology, we did assign a specific yield, hydrogen yield. Then we also looked at seafloor porosity, which is like, again, one of those fundamental inputs for the calculation method. And we also took to NOAA data set of sediment thickness to really be able to like have like a total budget of hydrogen production across all marine sediments. And this is what we came up with. So, I mean, not came up, but like this is the result of that calculation. So on a yearly basis, across all marine sediments, you produce about 7 teramoles. So that's 10 to the 12 moles per year. And on an electron equivalent basis, so if you convert that to electron equivalents, this approximates to about like 1/10,000 of the production of organic carbon in the like overlying ocean water through like photosynthesis and carbon fixation. So hydrogen production from water radiolysis remains a pretty like small contributor to like microbial fuel on a global scale. However, like if you look at just like global organic flux to the seafloor, we get closer to 1 or 2%. So like radiolytic hydrogen. So that's like not completely insignificant. But now like the important like part here is that like, although like organic carbon fixation, like, I'm sorry, I'll interrupt you and then that's for the next topic. So the last question, which is like really looking at like the contribution of water radiolysis, so all those chemical compounds being produced, how relevant is that process to subseafloor microbial metabolism? So only looking at marine sediment, not at the overlying ocean, just like really focusing on like the subseafloor. And the result of this analysis really show that water radiolysis is the principal source of biologically accessible energy for microbial communities in sediments that are older than a few million years. And I'll show you how we got to this observation. So here again, like the pretty like simplistic breakdown of water radiolysis. So water that decomposes to hydrogen and hydrogen peroxide. Now, hydrogen peroxide is like, can be pretty detrimental to microbial communities, but it's also pretty reactive, especially in the presence of mineral, and those minerals catalyze in decomposition to water and oxygen. And oxygen is like a very powerful oxidant for like reduced compounds and not detrimental to microbial communities. So while assessing the relevance of this process for subseafloor microbial metabolism, it's really important to differentiate like water radiolysis as source of microbial fuel. So really only looking at the hydrogen, so as source of like reduced compounds that's able to be oxidized, versus water radiolysis as source of oxidants, so as source of oxygen and hydrogen peroxides that can be used for like the oxidation of organic matter, for example. And so the subseafloor can really be divided in like oxic and anoxic sediments. So here you can see like a map created by Dante and colleagues that really shows how like the like distribution of those zones. So blue zones are zones where like oxygen is present in subseafloor sediment from like the sediment water interface all the way up to the basement for the like dark blue, and in the lighter blue, like the oxygen doesn't persist all the way until the basement, but it's still like present for a significant part of the sediment column. So those are like really like oxic subseafloor environments where like organic matter is very scarce / inexistent because like it's all been like already oxidized. So in those blue zones, and really those oxic sediment, those are the zones where radiolytic hydrogen will be extremely relevant, because those sedimentary environments are really limited by organic matter. So they're really limited by photosynthetically like produced fuel for the microbes. While in all the white zones on this map, which are like the anoxic sediments, so those sediments are really rich in organic matter. And because they're so rich in organic matter, there is no oxidant left because it's already been all used up. And in those zones, that's where like radiolytic oxidants are extremely relevant because they're inexistent from like other sources. So we really differentiated our analysis in those two types of sedimentary environments, oxic and anoxics. Now, the first step is to really like constrain how much hydrogen is being produced downhole, like being produced, not only produced, but being consumed by microbes like downhole, because we already like kind of have a theoretical idea of how much is being produced. So for that, we looked at like actual measurements of hydrogen in both oxic sediment and anoxic sediment. So you can see the location of those cores. And on the left side, you have like 3 oxic sediment sequences, and on the right side, 2 anoxic sediment sequences. And the open circles are really like hydrogen measured in those cores. The solid line is like how much hydrogen is being produced, assuming that there is no biological sink or no chemical sink, just assuming like there is no sink for that like hydrogen. And you can see that there are between like 3 to 4 orders of magnitude discrepancy in the concentration of hydrogen between what's measured and what's expected if there was no microbial consumption. And that's true for both oxic and anoxic sites. So at this point, the most like one of the obvious way like this like expected hydrogen is not measured is true, like because there is like microbial consumption. So like this was kind of like our proof that like calculated hydrogen production rates downhole are most likely are a good proxy for like microbial consumption rates of that hydrogen. So that's like the figures we use for our analysis. Now, if we look at the other side of the equation, so really looking at total organic carbon downhole for like, here is an example of an oxic site in the South Pacific. We can really look at like how much like of that organic carbon is being oxidized. And so downhole by like, again, looking at the downhole profile of measured oxygen, and really like using a numerical method by Wang and colleagues that based on like statistical significant changes in the curvature of the oxygen downhole oxygen profile, we can like delineate zones of like microbial activity that are like statistically distinct from each other. And that's like the third graph on the right. So you can see that like you can approximate rates of like organic carbon oxidation based on that method. And so really looking at like radiolytic hydrogen consumption rates versus oxidation rates of organic matter, we can really like start looking at comparing scales of like processes, like is like organic carbon being like consumed faster, or is radiolytic hydrogen being consumed faster? So to like have an idea of that, we really defined a ratio of production / consumption of radiolytic hydrogen, again expressed in moles electrons per square meters per year, and divided by the oxidation of organic matter. So like really like both processes. And so when you take the ratio of that, if that ratio is 1, it means that both like the organic matter and the radiolytic hydrogen is being like oxidized at the same rate. And then we can kind of say like, well, both processes have the same relevance in terms of like sustaining those microbial communities. But if we have a ratio higher than 1, that means that like radiolytic hydrogen is like a primary source of like fuel for those microbial communities. And here you can see like a result of this analysis for various sites, both and various sites like in not only in across various ocean basins. And you can see the evolution of that ratio with sediment age. And you can see that like in young sediments, that ratio is like lower than 1, really showing that organic matter is the dominant field for those ecosystems. And that's like, it's pretty intuitive because like in the upper sediments, like in young sediments, you still have a lot of organic matter. It's like not very recalcitrant because it's still pretty fresh material. Well, the older the sediment gets, the more recalcitrant the organic matter becomes, but like that doesn't really like impact the amount of radiolytic hydrogen production. Radiolytic hydrogen production, if your lithology doesn't change downhole, is pretty constant with depth. So that's why like the older the sediment gets, you can see a real transition of that ratio, really like showing that increasing relevance of radiolytic products to like sustain microbial communities with sediment age. Now that's like really an analysis looking at oxic sediments. So where like organic matter is scarce and where therefore radiolytic hydrogen is extremely relevant as source of fuel. Now, if you look at the other side of the spectrum, so more like the anoxic sediments where there is a lot of organic matter, but not a lot of oxidants to oxidize it, that's where like those are the zones where radiolytic oxidants will be relevant. And here we did a very similar analysis where we look at like how much like dissolved inorganic carbon is being produced. So we used DIC production as really your like measure of like organic field respiration, and we compare it to how much radiolytic oxygen production is like being consumed. And here the story is not as clear, but like your ratio definitely like is close to 1, sometimes higher than one, showing that like oxidants are like from radiolysis are like more relevant and like also your primary source of oxidants in those environments. So again, highlighting the importance of that process. So if we put everything together, this study really showed that like, first of all, like radiolytic production in marine sediment is like more elevated than what was previously known, like assuming like yields of pure water. So really showing that like marine sediment have like a catalytic effect on radiolytic production. Then we show that like on a global scale, radiolytic hydrogen production is a pretty small fraction of like, in terms of contribution of bioaccessible energy on a global scale compared to primary productivity, it's only a couple of %, so pretty small, like pretty negligible contribution. However, if we only focus on the subseafloor, both hydrogen as well as radiolytic oxidants are really principal sources of biologically accessible energy for microbial communities. So this not only has implications for our understanding of life in the subseafloor on Earth, but also like for like assessing habitability of other planets and other subsurface environments on this planet. So it's a pretty significant shift in our understanding of like sources of energy and how those microbial environments are fueled. So with that, I would like to thank all the oceanographic programs that provided samples, as well as all my colleagues and contributors on this study. And I'll be happy to take questions. And if there are any follow-up questions later, I added my email address. So thank you very much for your attention.
I have a question. Do these processes, are they at all impacted, say, by like earthquakes or major shifts in the sediment structure on the seafloor at all?
Not really, because like even after like an earthquake or a shift like what you described, like the mineralogy or sediment like characteristics won't really be impacted in terms of like, I mean, the same level of radioactivity will still be present in like the marine sediment, like won't really like impact like the fundamental parameters like driving like production, for example. However, like earthquakes and so on, like do release hydrogen, like a different source of hydrogen. So you'll have like extra input, for example. But that's like completely like separate from like water radiolysis.
Justine, I had a question about the oxygen that's produced during radiolysis. So in an anoxic environment, if you produce that oxygen, are there microbes there ready to that can utilize that, or is that going to, say, oxidize some reduced iron or sulfur first for that type of community?
That's like, yeah, that's like definitely something to consider and like kind of an issue in the conceptually, but I would feel like, yeah, I think at this point I don't have like enough information to be able to like give you like an accurate answer, but like that's definitely like it could also be utilized to like oxidize the surroundings for, yeah, obviously. So yeah, I'm not sure how you would like actually like differentiate that process or like, yeah.
That's cool, yeah, because I'm just confused by some of these cryptic iron and sulfur cycling. You know, there's evidence of it, but there's no,
Yeah.
you don't see pore water profile. So it's really exciting to see, you know, how abundant this process is.
Yeah. Good to see you.
Yeah, thanks for a great talk.
Someone in the chat also said, fascinating talk, thanks.
Thank you.
Any more questions? If not,
Oh no, Steve stuff. Okay.
okay. So if not, thank you everyone for joining in, and thank you again, Justine, for giving us a really awesome talk.
Thank you. Have a good day.
Have a good weekend, everyone.
Bye.
Happy Halloween.